Hi, I'm Rezvan, and I'm going to be talking about a hierarchical model that we've been working on to get resting state networks at both individual and population level from very large-scale fMRI datasets. In recent years, the increasing availability of big fMRI datasets, such as UK Biobank, has enabled us to get more detailed mappings of the brain function and disorders. In particular, it has enabled us to have more detailed functional and network parcellations of the brain, and use those parcellations to estimate functional connectivity and build connectomes in populations and individuals. And finally, to use the output from those two steps to try and predict cognitive function and brain disorders in individual subjects. As a result, there has been increasing interest and lots of efforts into getting hierarchical models that allow us to estimate functional brain networks, not only at group level, but also at individual subject level. And the available algorithms, oftentimes, they either start from a fixed group model and they estimate subjects based on that group model, or they start from single subject modeling and combine those to get a consensus group level representation. More recently, there has been also efforts into getting hierarchical models that allow for bidirectional flow of information between subjects and group, and therefore they can give us more flexible and accurate estimations at both levels. One example of such models is Profumo, or the framework of probabilistic functional modes, that was proposed by Sam Harrison and colleagues a few years ago. The main idea of Profumo is to use Bayesian inference for getting a hierarchical model that allows for top-down regularization of the network parameters using group-level priors, as well as bottom-up regularization of the network parameters using single subject decompositions. It then combines evidence across all the subjects, accumulates and combines evidence across all the subjects to get an updated version of the group. And by iterating between subject level and group level representations, it continues until convergence. So the framework is flexible and it's powerful, and we can use it to estimate networks from both task and resting state data. Here we focus on resting state networks, or RSNs, for simplicity. However, the model is computationally expensive. More specifically, if we want to use it to apply it to thousands of subjects in UK Biobank, we start requiring tens or even hundreds of terabytes of computer RAM memory, which is currently intractable. Therefore, in this study, we wanted to continue the same line of research as Profumo and get a bidirectional hierarchical model of RSNs, with the key requirements of our model being scalable to very large population data sets. For example, UK Biobank is expected to have 100,000 subjects eventually. Additionally, because it can be expected for these large epidemiological studies to have heterogeneous populations, we want our model to be able to account for that kind of heterogeneity in the data as well. More specifically, through single subject modeling, we want it to be able to account for cross-subject variabilities and therefore increase our ability to predict cognition and behavior from the RSNs. Therefore, we propose an extension to Profumo, and we base our model on a stochastic variational Bayesian inference. Right, so stochastic Profumo. The main idea here is to replace the classic variational inference with the stochastic variational inference that entails visiting subjects in small batches rather than the entire populations of subjects per model iteration. However, we don't want this randomization process to compromise the accuracies of our estimations, and so we take extra things into account. Firstly, in order to make sure that our group model remains representative of the entire population, we treat group level parameters as global parameters and we update them continuously. They also keep a memory of not only current batch, but also all the previous batches that we have visited. Secondly, in order to make sure that all subjects remained aligned with the most updated version of the group, we always estimate subject-specific posteriors using group-level parameters as priors. And finally, in order to make our model more flexible to the heterogeneous population, every time we revisit a subject, we build its model based on previous incarnations of the subject model itself. And by making these changes in the model, we were able to decrease its RAM requirements quite significantly, so that if we want to apply it to 100,000 subjects in UK Biobank, this can easily be done using available compute servers. Right, now that we have the model, the first step was to evaluate its performance using simulations. We simulated 2 data sets, each consisting of 500 subjects, and they had realistic fMRI time courses and noise levels. Our simulations were specifically focused to evaluate the performance of the model in accounting for cross-subject spatial variability, and we compared the stochastic Profumo to classic Profumo and a spatial ICA, which has been a common approach in the previous literature. We used FSOS melodic as a measure of as a model of a spatial ICA at group level, and melodic plus dual regression for single subject estimations. And finally, we evaluate the accuracies of all 3 models at both group level and subject level. Right, so here are the results. So at both group level and subject level, when we compared the classic and stochastic Profumo, we saw that their performances are similar, and they both outperformed ICA paradigm at both group level and subject level. More specifically, at group level, they were on average around 20% more accurate than spatial ICA. And at subject level, they were the difference was even more pronounced, with the Profumo models performing up to 100% better compared to the melodic dual regression paradigm. So the single subject modeling in the Profumo hierarchical framework allows us to account for more cross-subject variabilities. Right, the next step, now we applied the stochastic Profumo to 5,000 subjects from UK Biobank. The data was pre-processed using a standard biobank pipelines. We applied additional smoothing and data normalization, and then estimated 150 RSNs at both population and individual level. Our stochastic parameter updates included thousands of updates at group level, with each subject being updated 50 times on average. And here are all the RSNs that we found. So we had both distributed modes, such as default mode, and more confined modes, such as modes in the visual cortex. And when we look at all the modes, they cover different brain areas and giving us a high-dimensional soft parcellation of the brain. To have a closer look at the modes that we discovered, here are 7 examples of 7 modes in the visual cortex, the group level at the top, and 2 example subjects at the bottom. And we can see that there are qualitative differences between subjects. Here is another example of 3 variants of default mode network, with the group level on the left and so 2 example subjects on the right. We can see that the modes are largely overlapping, but there's also lots of spatial detail differences between them. Right, now that we have the modes estimated, we wanted to look at the spatial and temporal network matrices, connectivity matrices, to see how independent or dependent they are. This is the spatial correlation matrix, or a spatial netmat. And we found that based on spatial correlation, we find 6 distinct clusters. They were in the somatosensory, visual, default mode type, parieto-occipital, frontoparietal, frontocentral, and subcortical brain regions. In spite of the modes being spatially highly correlated, when we looked at the temporal correlations, the matrix was much sparser, meaning the modes were much more independent. So the modes that we discovered were spatially correlated and temporally independent. Right. The final step was to see the performance of our stochastic Profumo model in predicting cognitive tests in UK Biobank and compare it against melodic dual regression. Our prediction paradigm combined different elements of both models, including a spatial layouts of the modes, as well as a spatial and temporal correlation matrices. We used each RSN to predict cognitive outcomes, and there were 68 cognitive tests from UK Biobank that we used here. And here are the results with the stochastic profumo on the y-axis, dual regression on the x-axis. And as we can see, particularly for best performing modes and cognitive tests, profumo's performance is superior to that of the dual regression's performance. And here are some of the best performing modes from the stochastic profumo for prediction of cognitive tests. They are both distributed, such as DMN, or confined, such as this visual network. Okay, so to conclude, a stochastic profumo allows us to infer RSNs at both individual and population level from UK Biobank. And because of a stochastic inference, it allows us to model more heterogeneity in big data populations. When we compared it against melodic dual regression, using simulations, we found that it's more accurate in estimating cross-subject variabilities. And when we compared the two models for prediction of cognitive tests, we saw that profumo's performance was better. So the single subject modeling in a hierarchical framework helps us here. Right, this is it. I'd like to thank our team, especially Sam Harrison and my supervisors, Steve Smith and Mark Woolridge. And thank you very much for listening.
Hey, welcome to my talk. My name is Jacob Billings, and today we're going to be talking about some topological methods to inform functional connectivity dynamics and to help decode brain states. So if we think about states of the brain, some previous studies have shown that we can look at the bold activations themselves and pretty well decode focal brain states at particular points in time during particular tasks. This is across 100 volunteers or so looking at HCP data. But one of the problems with this particular analysis is that if we're just looking at bold activations, the assumption is that all of these regions of the brain, these parcels, are independent from one another. But the brain is a complex. And I mean that in the verb tense. It is the case that if a system builds itself as multiple independent parts. Sorry, I'm having trouble moving to the next slide.
Hi, I'm sorry. What's going on?
Yeah, I was having trouble moving from this slide. It was working just a moment ago. Ah, here we go. Okay.
Okay.
I think it was the fact that I had my stylus on the thing. Is it okay if I start from the top?
Yes, please. That would be perfect.
Thanks. Sorry.
No, don't be. Trust me. You're good. All right, ready?
Yeah, thanks.
Yeah, of course. 5, 4, 3, 2.
Hey, welcome to my talk. My name is Jacob Billings, and today we're going to be getting into some topological methods to inform functional connectivity dynamics and help to decode states of the brain. In previous studies, we were able to discern that simply looking at the bold activations pretty well resolved brain states over the course of time at particular instance. This data is from the Human Connectome project. And what you're seeing is about 100 individuals and the embedding of the brain state similarity matrix into 2 dimensions. And as you can see, at particular points in time during the scan, you get a pretty good focal group structure as far as what the brain is doing at that particular point in time. But the problem with this analysis is that the brain isn't really a set of independent nodes. It's a coactive complex. And what we want to think about, rather than simply the bold activations, is the complexing of all of these computational devices that form the central nervous system. But how do we do that? How do we understand this high dimensional data set Well, some of the methods from topological data analysis are really great, very great for understanding a system in terms of its qualitative geometric information. Topology really is the study of connectivity information. So in our talk, we're going to go through some pretty traditional points, but we're going to focus for the most part on comparing time-varying functional connectivity metric spaces. That is the similarity between instantaneous points in time and the graph structures that are measures of the brain state at that point in time. So the data for the study has been used a couple of times. And the reason we're using this is because it's a continuous bold scan for 25 minutes. And a series of experimental stimuli were delivered: resting state, a memory task, video task, some mathematics tasks. And so we can look at the brain state across here 18 individuals across the same set of tasks in a continuous scan. For our time-varying functional connectivity matrix, we're going to be using wavelet coherence. Now, in our case, we're using a mean weighted or a weighted average over the spectrum of the wavelet coherence. And we're doing this simply for computational reasons, so that instead of having a multi-graph of time-varying connectivity graphs from each spectra, we concatenate that into a single mean average coherence graph. And the weighting is with respect to the cross wavelet power. So some of the graph metrics that we're using are pretty traditional. We're using a weighted Jacquard distance between the value of the graph edges themselves. As you saw before, it's just the activity of each of the regions of the brain. The strength of these edges, which is essentially the weighted degree of the time-varying connectivity graph. So as the alternative, the topological approach, it's useful to first look at some definitions of what topological features are. We can think about connected components that form a clique. We can think about a complex of elements in multiple dimensions of these connected components. In the zero-dimensional case, it's the nodes. In the one-dimensional case, you're looking at edges. You're looking at surfaces, solids in higher and higher dimensions, etc. And if you think about a series of connected nodes that are only connected in a cycle, then at least in the two-dimensional case, there's a hole. In the three-dimensional case, you have a void around the cycle of surfaces. So when we're thinking about weighted graphs, if we want to identify the shape of the multi-scale structure of this high-dimensional point cloud, we can do what's called a filtration across these edges, which looks at the incremental addition of edges, surfaces, etc., in multiple dimensions, as you add these simplices to one another across increasing distances. And you can log then the occurrence of these holes. They're called homologies because regardless of how you distort and bend without tearing the relationship between nodes, these voids are going to appear in all of those bendings and stretchings. They're homologous across slight variations in your data structure. We can plot the birth time of these homologies and also the death time as the holes are filled in. And if we plot the system, we can end up getting a 2D point distribution that is amenable to comparison using an Earth mover distance or a high-dimensional version of that, a slice-Wasserstein distance. Here are some of the definitions I just went through, and you can look at those as you wish. So once we've got this metric space from the simplicial-based approaches or the homology-based approaches, we want to embed them into 2 dimensions in order to better visualize what's going on. One of the concerns of this embedding is that it's fundamentally transductive, which means that if you've got really similar points in your high dimensional space, once you embed them into the low dimensional space, you're going to preserve that high dimensional structure in a very granular way. So if there's a very strong connectivity point to point, you're going to preserve that in the low dimensional structure, and you might get this string look to your plot. If you do some subsampling by half, you might end up seeing that volunteers really cluster very close together. So if you really want to subsample a lot, you can get a point distribution that references out of sample points. And those out of sample points will give you a nice cloud that observes the relative structure with reference to these reference points. So here are our results. The rows A and B are simply one realization of this embedding. And row C is a realization of this embedding from 64 randomizations of our initial points. In row B, you see that there are some colorations added to the density map. And this is a watershed distribution of the relative density of points across this embedding. We use this watershed distribution to identify very dense regions that are that the negative of the density map form catchment basins, really low points. And so we can do some great clustering on a two-dimensional representation of our data. So if we look at these clusters, the first thing we want to understand is how generalizable our embedded distribution is across volunteers. And we can do that by doing a statistical test for the number of volunteers in each clusters that are not insignificantly represented in that cluster. And so we want to see a distribution that's shifted to the right. The numbers that you see in row C on the top are a weighted sum of that significance test. So the number of points times the number of volunteers that are not insignificantly represented is this weighted sum. And as you can see, the embeddings from homology outperform those from the simplex-based approaches. And that is to say, the homology-based approaches are more generalizable in terms of how the points distribute. The points that are representations of the state of the brain distribute across this embedding. There's greater overlap across volunteers. Now we can do the same thing with looking at the cluster-wise segmentation in terms of the experimental stimuli that are given. And row C is again this average from 64 randomizations of the statistical significance of finding a stimuli in a particular point. Now I should mention that all of these statistical tests are Bonferroni corrected. Again, it is the case that the methods from homology are performing really, really well. In particular, the diagram 1 distances, the one-dimensional homology groups, perform the best. It's also the case that video tasks tend to be discernible really well with all of the different metric spaces, while the homology-based approaches really outperform in terms of the mathematics task. So if we think about these clusters that are significant for particular stimuli, we can also do significance testing with respect to the performance of each individual in that particular task. And that's really the takeaway from this particular slide, is that we're doing this significance testing. But what does it all mean? You know, what is the case of seeing a group of points that are significant for a particular kind of performance, higher or lower correct scores, more or less time taken, more or less missing values? Well, we can get a sense of that by, for each cluster having significant performance, taking the mean graph of the points in that cluster and then comparing those mean graphs across different embeddings with different performance. And this comparison is just the weighted Jacquard distance. As you can see, most often the comparisons across methodologies, most often the comparisons across task performance show stronger difference between these mean graphs when using the embeddings from homology. That is to say that the mean graphs from homology show greater difference in terms of the brain structure conducive to different types of performance. And we can look at the top 10 strongest edges in this graph And they're pretty much the same between the diagram 1 metric space and the simplex metric space. But if we look at for each node, the mean edge difference from that node, we can see that the difference between these mean graphs is stronger node-wise for the diagram-based approaches than from the simplex-based approaches. And again, this reinforces the notion that the embedding space from homology is better distinguishing differences in performance, differences in stimuli, and also is more generalizable across volunteers. So, this isn't a very great visualization for homology, because homology is really giving us an impression of a quantification of the shape of the data. And we should probably move past this idea that the brain connectivity is simply measured by edge weights, and that the strongest edges are going to be really significant in terms of what's actually going on in the state. If there is a weak connection between regions, that's significant too. If there's a cycle forming a hole inside of the brain, that's also a really interesting feature. And we want to build some visualizations that show that. Some of the work that is being done now is really trying to captivate this topological inquiry into brain complexity. And with that, I'll conclude my talk. Here are some references if you want to know more about what's going on with any of these subjects. Thank you.
Hello, everyone. My name is Nan. I'm a postdoc from Georgia Tech and Emory Biomedical Engineering. I'm currently working with Dr. Shaila Kaiholtz. Today, I'm going to talk about my project, which is to investigate the spatial temporal functional interactivity among the large-scale brain networks. So in this talk, I will briefly talk about the background and the problem, and then describe the method we recently developed to solve this problem. The method is called prediction correlation, or just PCOR for short. And then I will demonstrate some key results and conclude my talk. So here is the brief background of the resting state fMRI analysis. As we all know, the correlation method has been extensively used to measure the resting state functional connectivity. In the last decade, there has been substantial efforts being made to identify the spatial temporal structure of this resting state functional connectivities and networks. There are two important lines of work. The first one is many computational methods have been developed to estimate the directed or the effective connectivity. And in parallel, some other studies have also been done to review that the correlated yet temporally asynchronous patterns of both signals actually reflect the timing of the information transfer in the brain. So all this together would allow us to identify not only the directed spatial properties, but also the temporal influence in the network architecture. So in this project, we leveraging a recently developed analytical approach called prediction correlation by us, and investigate the causal interactions, which include both the directed spatial property and the temporal influence between the regions among the large-scale brain networks. So the prediction correlation, the computation roughly involved two steps. If we consider all ordered pairs of both signals, X and Y, the first step is to predict the duration of the information transfer. So specifically, we compute the prediction of Y based on X through a time linear invariant causal model as characterized in this equation. And this NXY is the time lag, which is optimized by minimizing the prediction error through the information criterion. Then the duration of the information transfer, D, it's computed as the time sampling rate times the time lag. And the second step is to compute the strength of the directed functional connectivity. this is simply computed as the correlation between the signal Y and its prediction. And at the right, I listed several key features of the p-correlation. So the first one is that the p-correlation can estimate a full directed functional connectivity map and a full duration map for all pairs of ROIs. And the second one is that it is pairwise asymmetric. As we see in this flow chart, if we simply flip the two signals X and Y, we'll get different duration D and different values for the p-correlation rho. So that means they're asymmetric for in both duration and the strength. And the third feature is that if we restricting the duration to be one sample, then the p-correlation just gave the correlation result. And the fourth feature, which is a very important one, is that the p-cord map is able to capture the modular organization of the functional networks. What does it mean? So basically, if we cluster the directed FC map that is estimated by p-cord, we'll get exactly the same set of functional map as if we cluster the standard correlation FC map. So all these key good features allow us to do some new investigations on the spatial temporal functional connectivities. The first one is allow us to look at the long versus the short duration of the information transfer. And the second aspect is to look at the pairwise asymmetry in both the strength and the duration of the information transfer. So we test the p-correlation methods on the HCP 500 data. We randomly select the 200 subjects. So it has a TR equal to 0.72 seconds, and we extract the signals from the Gordon parcels. And here, I will show some results. The first set of the result is the long duration directed FC, and these two figures just show that the full directed FC map and the full duration map that is estimated by p-cord. And as far as we know, no other methods can compute them efficiently thus far. So the long durations are closely associated with the strong functional connectivity with the correlation equal to 0.72. And then we look at where they're located. And we can see in this duration map, the long durations are mostly dominated by the within network connections, within like in the somatomotor, visual, and visual system, as well as the information transfer from the somato to the visual systems. And next, let's look at some short duration directed FCs. So we determine the short duration information transfer by just looking at the lowest 4% non-zero values in this duration map, as well as looking at the p-correlation with p-value smaller than 0.005. So the second condition it's to reduce the low value FC, which is due to the noise. And as we see in these two figures, the short duration, in contrast to the long duration FCs, it demonstrates a hub-like structure. What do I mean by hub-like structure? So we can see that there's dense dotted rows and columns in these two maps. And the rows are actually the high degree outward hubs, which are the ROIs that are actively sending the information from itself to the rest of the cortex. And the dense columns, we're calling that they are the inward hubs. Those are the ROIs that are actively aggregating the information from the rest of the cortex to itself. And we can see that the outward hubs have higher degree than the inward hubs, like have a denser dotted lines. And then our immediate next question is, where are these hubs located? And then we map those hubs on the brain cortex. And we can see that the high degree outward hubs, which are actively sending the information, are mostly located in the visual and default mode system. those hubs are consistent with the previous funding of functional hubs, but we provide some additional information on the information transfer duration, both in the duration and spatial direction. And the moderate degree inward hubs are primarily located in the hippocampus. That seems to indicate that the resting state are supporting the memory consolidation process, which is actively aggregating the information from the rest of the cortex. And as we see for both inward and outward hubs, they're bilaterally symmetric, which seem to be very reasonable. And then the third set of results is to looking at the pairwise asymmetry. Before I go into the details of the numbers, I would like to show you a brief example on how this pairwise asymmetry may look like in brains. So here is the default network, the top strongest connections within the default networks, and the connections going into the default networks, and also the information going out of the default networks. In all the three figures, there are arrows that are pointing out the direction of the information flow, and the whiteness of the arrow is the strength of the functional connectivity, and the color represents the duration. So the warmer the color has, the longer the duration the information transfer has. So as we see that the inward and outward information transfer, we can see some difference in the strength or in the arrows, but there are more obvious difference in the color, which is the duration. And overall, we found that there is significant inward-outward difference in duration, but not so much in the strength. That's almost for all pairs of ROIs and pairs of network interactions. And then we look more details on the pairwise asymmetry in the duration. And this figure shows the average duration of the information transfer coming from, which is the outward, and going into this inward, each of the 333 Gordon parcels. As we see that they are actually plotted in the same color map, but we see there's definitely obvious distinctions in the outward direction and inward direction in the durations, and they are also quite bilaterally symmetric. For example, the visual precunions shows long inward connections with short outward connections, like this region. But in the contrast, the limbic system has the reverse pattern. And on the other hand, the prefrontal cortex has roughly similar inward and outward connections in the durations. And here's the conclusion. We use the recently developed method called prediction correlation to investigate the spatial temporal functional interactivity in the large-scale brain networks in resting state. And for the first time, the information transfer between the remote ROIs in both the spatial, which is the direction, and the temporal scale was characterized. So there are two central observations. The first one is that the short information transfer are driven by multiple brain hubs. And the second observation is that the duration of the information transfer is asymmetric between the inward and outward directions. Our discovery is consistent with the current understanding of brain function, but provides some novel insights in the spatial temporal scale. So for this project, I would like to thank my advisor and collaborators, Dr. Shaila Paiholz, Dr. Nathan Spring, and Dr. Peter Dorshak. So without their help, I cannot complete this project. We have a paper, a manuscript that's been archived and currently have been submitted and reviewed and currently under revision. So welcome for any questions or discussions. Thank you.
Hello, everyone. I am Zhang, a PhD candidate from Fudan University and also a visiting researcher at University of Cambridge. Today, the topic of my presentation will be the brain gender spectrum. Briefly speaking, we aimed at constructing a continuous gender through brain functional networks. Sorry. Here is the overview of my presentation, including the background introduction, methods, and the demographics, 3 major results of our study, and finally, a conclusion. As we all know, biological sex or the genetic sex is binarily determined by sex chromosome. But in many psychological studies, starting from Sandra Bam's study published in 1974, suggested that human gender should be described by a continuous sex role rather than the binary sex. In this framework, androgyny was associated with better mental health, since the androgynous person possesses both feminine and masculine traits without employing a gender schema. But such theory lack brain evidence. What we want to do now is to construct a continuous gender using brain functional network. Here is the pipeline of our study. First, we studied sex differences in global and local functional connectivities. Then, based on those sex differences, we trained an SVM classifier on UK Biobank participants and reached a 10-fold cross-validation classification accuracy of about 80%. We also conducted the sensitivity analysis of our classifier. We tested multiple confounding factors influence to our model to ensure the reliability and the validity of our model. Then our model was tested on an independent dataset, the HCP dataset, and reached a classification accuracy of about 77%. The likelihood that a given brain functional network to represent a male brain is defined as brain gender spectrum in our study. And the androgyny was located in the middle part of the spectrum. Finally, we applied our brain gender spectrum into internalizing problems. The internalizing score was composed by anxious, withdrawn, and somatic complaints. Here is the demographics of subjects used in our study. Large cohorts, including imaging for adolescents, Human Connectome projects for adults, and the UK Biobank for elders was used in our study. Also, we used the data from National Yangming University. It is a relatively small dataset, but it includes participants aged in different age periods. The mean frame-wise displacement smaller than 0.3 was set as a threshold to ensure the quality of our data. Here is the first result of our study. We first studied global sex differences. We found that global sex differences changed non-linearly with age. The global functional connectivity increased during younger period and decreases during older periods. And the trajectory was not confounded by covariates such as mean frame-wise displacement, total intracranial volume, and the signal-to-noise ratio. The global connectivity evolved differently in female and male subjects, and those two trajectories intersected at about 45 years old. To ensure that our findings was not confounded by side effect, we also used the YMU dataset as a validation. We found that the YMU dataset produced similar results with that of multicenter data. Female trajectory and male trajectory also intersected at the age about 45 years old. In this figure, the trajectory was fitted by a localized spline curve. After studying the global sex differences, we also studied local sex differences, both at network level and at regional level. we calculated the effect size of sex differences in 3 datasets respectively, and we found that in younger subjects, salience network had the most significant sex differences, but in older subjects, the default mode network had the most significant sex differences. Then, at the regional level, we found that cingulate cortex showed the most significant sex differences in younger adults, but the angular and precuneus show the most significant sex differences in older adults. And based on those sex differences, we trained our SVM classifier on UK Biobank dataset and the test on the HCP dataset. The test accuracy is about 77%. We also tested the influence of confounding factors like TIV, SNR, and MinFD to our model. We found that those covariants related to image quality, like signal-to-noise ratio and mean frame-wise displacement, won't influence the performance of our SVM classifier. Then we conducted the sensitivity analysis. We tested the influence of pixelation, also the training set sample size, the age configuration of training samples, and the brain networks used in our model and those influence to our model to ensure the reliability of our classifier. Finally, based on our classification model, we applied the gender spectrum to internalizing problems, and we found that internalizing scores was quadratically correlated with gender spectrum, and the androgynous people have least internalizing problems. This significant correlation was most prominent in male subjects. Following that, we also studied the 3 subscales of internalizing problems, and we found all those subscales were significantly quadratically correlated with gender spectrum. So, as a conclusion, we first studied sex differences, and we found that global functional connectivity changed nonlinearly with age, and the trajectory evolved differently in female and male brain. Then we found that salience network and the default mode network had the most significant sex differences. Based on the sex differences, we then built a reliable SVM classifier and establish a continuous brain gender using the classifier. We found that the gender spectrum given by the SVM was quadratically correlated with internalizing problems. So that's all for my presentation, and I want to thank many thanks to my supervisor, Professor Jianfeng Feng, and my instructor, Dr. Zhang, and the members of ISTBI at Fudan University, and also my supervisor, Barbara Sahakian, and the members in the Department of Psychiatry at the University of Cambridge. Thank you for your